Hello everyone, in this poster presentation I will be bringing you a little bit about my research work at the Brazilian Laboratory on Silence Technology located at the Universidade Estadual de Campinas, which is entitled Prospecting for Essential Genes in Meloidogyne Incognita through a multi-omic approach for silence by RNAi. Today, a large number of crops worldwide are affected by rock-nut nematodes. They are considered to be most widespread and destructive plant parasites nematodes and can cause an estimated wide loss of 25 to 50% of wide areas of cultivated land. Many measures that are not environmentally correct are being used to control this species. As an alternative, research done with RNAi interference technique has proved to be highly significant in the control of the agricultural pests. This is possible because this technology allows us to regulate the gene expression in order to silence its activity. As we want to exterminate or prevent the parasite-host interaction between the nematode and the plant in pest control, we look for silence genes which are essential, that downregulate the animal cannot either survive, develop to the next stage of life, or reproduce. With the immensity of data from the omics that we have available, the participation of bioinformatics is fundamental to manipulate and filter this data so that in the end we arrive at a list of genes that met our criteria to be considered essential. In this sense, our pest under study is the rock-nut nematode Meloidogyne incognita, which causes high damage to agriculture in Brazil and throughout the world. Here's a photo of a cultivar infected by the pest M. incognita. This pest uses the nutrient reservoir of the roots to ovulate, feed, settle there in addition to developing for the next stage of life, which ends up generating this appearance of galls in the root. The main goal of this work is to develop a genome and transcriptome data pipeline that returns a final list of essential genes of root nematode parasites, with M. incognita as the center model. For that, we need to have some gene selection criteria for being considered important for M. incognita to survive. It needs to be significantly expressed in J2 life stage, which is when the formation of galls occurs. Single copy orthology, because that's how we avoid functional composition between genes, and we can have a phylogenetic validation of this and conserve it between different cultivars of M. incognita. We believe that these features indicate that the gene is fundamental for this nematode. In our methodology, I formulated this illustration of our pipeline. We started by downloading the raw data from public databases, and these were 15 RNA-seq samples from M. incognita with 3 replicates for each of the 5 life stages, egg, juvenile 2, 3, 4, and female. We also downloaded 11 samples of nematode species well described in the phylogenetic literature of the genus Meloidogyne. Here is the caption of this pipeline showing that each pipe represents a data, and the arrows are the transformation that is performed with this data, and the software used in each transformation is indicated above or below the arrow, while the dashed arrow with the file at the end indicates a statistical analysis. It is separated into assembly quantification filter with the final notation of the resulting gene. Now that we have the transcriptome ready, we quantify our transcripts for each life stage, obtaining a count matrix for each transcript. For this matrix, we perform the clustering of our genes based on the expression behavior that their transcript presents. Thus, it was possible to distinguish the expression pattern of M. incognita genes in 12 different clusters. Having this information, we select only 3 of the 12 clusters because they showed an expression behavior which genes were differently expressed in the J2 life stage. With that, we filter our list that contain the entire genome of M. incognita to 10,153 genes, which would be all genes present in these 3 clusters. An enrichment analysis by genetic ontology was also performed. In the end, we saw that only the genes of cluster 4 were enriched. That is, they had their GOs overrepresented with some biological function. Next, we did the analysis for our second filter, which would keep genes that are orthologous in single copy. We selected then 11 species, 5 from the Meloidogyne genus and 6 other more distant nematode species, and searched for orthologous genes. In the end, we obtained 343 genes that are orthologous and have a single copy in common with the selected species. From this, we cross our current list of genes with this another orthologous list, which returns in 54 genes, which are different expressed in J2 and single copy orthologous. In our last filter, we look for genes that are highly conserved among M. incognita samples that come from different regions from Brazil. We manipulate data from a VCF file which contains observations of mutations in 11 specimens of M. incognita. We want to select genes that had a maximum of 5% mutation in relation to the total gene size for all cultivars. With this, we verified that the most conserved genes in incognita were those represented by these green bars in the histogram, which left us at the end with a list of 28 genes, which are bias expressed in G2 life states, single copper autologous, and highly conserved. We are currently carrying out silence assays in C. elegans with genes from this nematode that are orthologous and correlate with the genes of M. incognita that were selected at the end by our pipeline. We believe that by doing these assays, we will experimentally validate the efficiency of our pipeline in the research for target genes. We intend to do this test with M. incognita as well, but we are first in this pilot experiment with C. elegans to validate some parameters. In the future, we intend to apply our pipeline for prospecting optimization with biological data in addition to nematodes, with different other agriculture pest species, and expand this technology beyond the scope of this project. So thank you very much for your attention and have a great conference.